Now, to talk about the different stages of cancer, I want that before, when I studied biology, they show these scary pictures. Yeah, and now we can see also scary but pretty all these different behaviors that are related to cancer cells. And I later will show you these very pretty cells that are moving and avoiding obstacles. So remember, this is in a culture, so all of them will be in vitro, and the tumors that I generally, and I will show you if you have seen the work of Dagem, they are in vivo. The organism was alive and we saw the tumor growing. This is in a plate, so remember this will be in vitro, while the one that Dagem did will be in vivo. Okay, so why show you these pictures? Why show you these movies? Because you think, oh my God, this happened during cancer. Of course, I have never seen a cell behaving like that, and this showed that the cells are abnormal. And actually, all these behaviors are seen when you study developmental biology. You see that all these things had happened in different stages of development. Yeah, the vision, the appearance of this action protrusion, this single migration, all these things happen. Yeah, so all these cancer behaviors, cancer movements, it's not that they are abnormal because it's the first time that we see them. No, it's because we have seen them before in different stages and in different type of cells. What a cancer is doing is reusing the same genes in a different context, reusing different cell movements, but now for a different purpose. Okay, that is is a to some degree an evolutionary process that the cells are surviving and escaping to look for more nutrients and moving into a different tissue. So we what we see is uh, uh, that the evolution is speeding up because there is, as you saw in the with the HeLa cells, that are very fast changes. So although I cannot show you a human evolving, I can show you in few decades the cells of a human evolving and changing of function just through these genetic changes. Okay, so human evolution is not possible to see for a human. Yeah, we don't live millions of years, but our cells we can definitely see that, and this is why cancer is so prevalent. And as I said in the beginning, uh, two out of five people in Canada will develop cancer in their lifetime, and one out of four will die. Okay, so now we're going to talk about the stages of cancer. Um, what are the stages in cancer? They could be many different stages, yeah. But let's talk about three main stages from a very general perspective, yeah. So we're going to talk about in the first one, when you have a normal cell, here you have, and we are going to talk about later about that, that here you remember epithelial cells generally, and the epithelial cells are located, they are communicating with other parts of the body. And here you have an extracellular membrane, a basal membrane, that later we will talk about that. And look what happened. The cells start proliferating without control, and look, they break the barrier that they have underneath, yeah, and they are moving into the wrong place. Yeah, so this is is when the primary tumor starts. So you have a primary tumor, the cells start losing control, and they move where there is food. They move into the blood, yeah, and they uh, go inside of the blood vessels, and they start traveling there. This process is very unlikely to happen. Yeah, there should be an accumulation. Yeah, so accumulation of mutations to produce the extra proliferation. Generally, the immune system kills, and there are many mechanisms. There's apoptosis, but when you start accumulating mutations, this happens. That a cell destroy this barrier is very unlikely to. You need more mutations. So this is why it takes so long. Here, the process of intravasation, sorry, when the cells go inside of the blood cell, again, this is very unlikely to happen. Many cells have to die to get into the blood cell and to be viable. So again, this way takes so long. And even more unlikely is one of those cells are able to leave the blood vessels, yeah, and go and find a new process, a new organ, and produce a colonization. That here you call this micrometastasis. That is you start finding a secondary, you form a secondary tumor, and here is when you start the tumor start growing and take over. Okay, so this is what you call metastasis. This take a long time, but when it's achieved, that is sometimes at this stage it become more and more difficult to cure because you have to kill the cells that have already evolved all these abilities. So you might kill this tumor, you might kill this tumor, but if you haven't killed it, and you can kill this one, you kill this one, you can kill this one, you can kill this one, you can kill all of this one, and you can kill all of this one. But if you still have this one that are the one that have the property to migrate, the tumor is going to come back eventually, yeah, or it will eventually accumulate again mutations to do that. So I told you there are, from a very general perspective, there are 3 stages of cancer. Yeah, 1, 2, and 3. So here you have a primary tumor, then it goes to the blood sample, and then it recolonizes and form a secondary tumor. Okay, so and this is what we call metastasis when you go to spreading to a different parts of the body. So at the beginning you have a primary site invasion, that is the place where the cancer actually starts. Second, in the circulation, cancer cells usually get into a bloodstream or the lymphatic system, and we're going to watch a video about that and how the cells move. And later, they go into a secondary site. That is when metastases happen, when the cancer is spread to distant parts of the body. Okay, so now let's start with this primary site invasion, and here we're going to talk about more from a cellular biology, from the anatomy of this. And so the first step in metastasis is the migration of cancer cells away from the primary tumor. So here you can see a schematic where you see a normal cell, and here you can see a cancerous tissue. So look, and the different characteristics, and some of you will hear words that maybe you will remember some of them from cell biology, other ones you will remember them from anatomy and physiology, and some of them you will say like, okay, I have never heard about those, so I will go step by step, remind you some of those things. So first, I want that you think about what happened with this. Like, these are the epithelial cells, yeah. And if you remember, there is a barrier here, the basement membrane, that is located here. Is the green that separates the epithelial tissue with the stroma. So here you have the stroma, that is an organ or a tissue that have a structural role. Yeah. So the stroma. Let's put it here in red. This will be the stroma. And here in blue, in this case, there is an epithelium. Okay, what else do you see? It become more cancerous epithelium. Is more organized or more disorganized? What about the fibroblasts and the immune cells? Generally, you see the fibroblasts. Sorry, here. Let's put it here. You see a fibroblast here. You see a fibroblast here. Yeah. But what about in a cancerous epithelia? You know, is it moving in a different direction? What happened with in the stroma? Do you see that the cancer cells, the epithelial cells, are moving there? And what about the alignment of the collagen? Remember, collagen, very important protein that is fibrous, that is in charge of giving a framework, a structural support. This is why there are more. So look the difference between this line, this alignment, and this alignment. Okay. So let's try to see all the things that we said. So the epithelia, the blue things. So let's delete this so it's easier. Remember the epithelia, what I show you in blue is that the epithelia is organized. So clearly you see it that in the normal is you see organization. What happened with the basement membrane or basal membrane is intact. So you will see the green things, the barrier is still there located. Okay. Third thing that the stroma, let's put it here. There is no epithelial and no cancer cells. So look, no epithelial, no cancer cells. And finally, the fibers of the collagen in the stroma, they align relatively aligned with the basement membrane. Okay, so just to, in case, I'm going to put a definition here. So here is the basement membrane. Here in green, basement membrane or basal membrane is a thin, fibrous extracellular matrix of a tissue that separates the internal and the external body surface. Okay, so this is a basement membrane. And just to remind you, a stroma is a tissue or organ, yeah, that has a function that is or structural for support, or yeah, or that has a connective role, like here, yeah, that is separating the epithelial cells. Here you have the structure that remain that epithelial together, and here you have these blood cells. Okay. Okay, now for the assignment, I want that you send me that you fill this. What happened when it's invasive? Yeah, and the epithelial. What happened when with the basement membrane? What happened with the stroma? And what happened with the alignment of these fibers of the collagen alignment? Okay, so stop the video if you want and write those things down. It's pretty easy here. You see that instead of having a mono layer, yeah, not all the epithelial have to be a mono layer, but in this case it is start in a normal case as a mono layer. Here you start creating this agglomeration of cells, accumulations of cells that are not organized, that start forming tumors. Okay, what happened with the basement membrane? So here clearly you have a hole, yeah, and you have not a clear definition between where is the epithelial finish and when the stroma starts. So it's perforated, yeah, and we have a specific enzymes that can break those basal membranes, and that we will study. The stroma, here in the stroma in red, if you see here, you have in the stroma you have a mix between stromal cells, the collagen, the immune system, immune cells, and here you have the cancerous cell. Yeah, and the same thing happened here in the epithelial. You can find different type of tissues and tumors, as we will study later. They are more than one type of cells. There is a combination of different type of cells. And here you see a realignment of the fibers. So let me delete this here, and here you see that they are moving. There you can find new alignment here. Okay. Okay, so let's keep talking about the primary tumor, and now about some of the genetic changes. So, I told you that it can be divided in three very general stages: primary tumor, circulation, and metastasis or secondary tumor or distant metastasis. And it could be subdivided. So here, when the carcinoma in situ, that is in the correct place, yeah, and then when the carcinoma start invading. So I show you the different morphological changes that happen in an epithelial or the anatomical changes at the cell biology. And in addition to that, they are genetic changes that need to take place. Yeah, so Here the cells, as we talked before, similar, and welcome to my cat that he decided to join the class. Here we have that the cells have to transform. So here there is no virus, but the cells suffer a transformation, and they decide to invade the surrounding tissue. Yeah, they also should be these initiation genes not only transform a cell, but also attract a supportive stroma. Yeah, so the stroma right now is located here, and if you see, there should be a degradation of the basement membrane. So if you remember here, you have enzymes that break that, and other changes include that the cells become mobile. They experience angiogenesis that we will cover in the following class. Angiogenesis is a process that is just that new blood vessels, yeah, will appear. So here to feed these cells, they will need to create a new blood vessel here. So this tumor keep growing. So some of them will move here, but other ones can say, we need that new blood vessels appear. Again, this is something very common. Every time that you have an injury or something like that, new blood vessels need to reappear. Yeah, here they're using the same genes to say blood vessels appear here because we need more food. But in this way, it's just for the tumor growth. And here, something else that happens is that there should be changes in the immune system. So if you see here, the cells, let me delete this. Here you see in blue the cancer cells going, and surrounding the cancer cells, you see all these immune cells. But in this case, the immune cells are not attacking um, they are not attacking the cancer cells, and they generally are destroyed, or they go through apoptosis, or they are degraded by the immune cells or by killer cells, a type of immune cells. Here they nothing happened to them. So here I show you four type of mutations, so four type of changes that need to happen for the primary tumors to not only start growing but also move away from the primary tumor. Okay, so now we are going to talk not only in terms of the morphological changes, but also what happened with the cell adhesion. Yeah, because if you remember, epithelial cells, and if you see here, the cells are properly attached to each other. Why? Because they have adhesion junctions. Yeah, so it's like when you have two Lego parts together. Yeah, they are, or when you have bricks together, they have cement in between them. So same way here, in between each one of these cells, there will be proteins that attach them. Yeah, so let's make here you have two epithelial cells. Here you will have a protein that is going to attach them. Yeah. So what they need to do to be able to invade is to remove this adhesion. So it's like I'm going to take this. Okay, I couldn't do it, but let's think that I just remove the adhesion that it was here. Now you don't have adhesion. And what is going to happen? Suppose you have the adhesion molecule here, you have the adhesion molecule here, but now you don't have a adhesion molecule here. So this one can escape and can invade our tissue. So changes in cell adhesion are also important. Yeah. So they should be at the attachment of the cell of the malignant cell from the tumor mass. And how it happened? By the reduction of intercellular adhesion molecules, allowing this malignant cell to have motility and enabling to penetrate to the stiff stroma and later to the blood vessels. Okay, so there are many changes, and this is another change that need to happen. Okay, so So summarizing, we just talked about the first step, the basic primary tumor, that it goes from a carcinoma, an epithelial, a transformation from a normal epithelium to a cancerous epithelium that occur in situ in the proper in the place where is the epithelium is generally located, and it become invasive. And what you need to do is to have to break the basement membrane. I told you that it need to start becoming disorganized, that there should be proliferation, that you start mixing cells with the stroma, and they need to start going in that direction, that they should be angiogenesis, that they should be reduction of the cell adhesion, and there should be um, a lot of this process happening. And to do that, you will need require multiple mutations. Okay, now let's go to the other two stages, the circulation and the distant metastasis, or when it goes into a secondary tumor. So here you can see the second stage, yeah, that is the process of circulation, where it went from the local invasion, but it hasn't arrived to it has not gone through metastasis. Yeah, so here they call these progression genes. So here you have the idea is that they have different functions from the, so these genes are different to the genes that you need to colonize a new tissue. Yeah, so these are different genes, and it need to allows them to go through capillary walls. So yeah, as you can see here, you need to allow them to go through capillary walls, and they need to survive to the new tissues that they are going to be. Yeah, so you need to survive here in the blood cell, you need to survive to the stroma, and later they will need to survive here in the new tissue. Okay, so those are called progression. And here I'm going to show you a series, a video that I found very useful that explain how um, cell moves, yeah, how this cancer moves and go through different regions, and they are able to adapt and to go through. So what it was supposed to be a non-mobile cell, that is generally an epithelial cells, they have this structure, this junction that don't allow them to move into transform into a new cell that has mobility and that can go through different very small spaces and invade different regions.
Begins as a solitary tumor in a specific area of the body. If the tumor is not removed, cancer has the ability to spread to nearby organs, as well as places far away from the origin, such as the brain. So how does cancer move to new areas, and why are some organs more likely to get infected than others? The process of cancer spreading across the body is known as metastasis. It begins when cancer cells from the initial tumor invade nearby normal tissue. As the cells proliferate, they spread via one of the three common routes of metastasis: transcoelomic, lymphatic, or hematogenous spread. In transcoelomic spread, malignant cells penetrate the covering surfaces of cavities in our body. These surfaces are known as peritoneum and serve as walls to segment the body cavity. Malignant cells in ovarian cancer, for example, spread through peritoneum, which connects the ovary to the liver, resulting in metastasis on the liver surface. Next, cancerous cells invade blood vessels when they undergo hematogenous spread. As there are blood vessels almost everywhere in the body, malignant cells utilize this to reach more distant parts of the body. Finally, lymphatic spread occurs when the cancer invades the lymph nodes and travels to other parts of the body via the lymphatic system. As this system drains many parts of the body, it also provides a large network for the cancer. In addition, the lymphatic vessels empty into the blood circulation, allowing the malignant cells to undergo hematogenous spread. Once at a new site, the cells once again undergo proliferation and form small tumors known as micrometastases. These small tumors then grow into full-fledged tumors and complete the metastatic process. Different cancers have been known to have specific sites of metastasis. For example, Prostate cancer commonly metastasizes to the bone, while colon cancer metastasizes to the liver. Various theories have been proposed to explain the migration pattern of malignant cells. Of particular interest are two conflicting theories. Stephen Paget, an English surgeon, came up with the seed and soil theory of metastasis. The seed and soil theory stated that cancer cells die easily in the wrong microenvironment. Hence, they only metastasize to a location with similar characteristics. However, James Ewing, the first professor of pathology at Cornell University, challenged the seed and soil theory and proposed that the site of metastasis was determined by the location of the vascular and lymphatic channels which drain the primary tumor. Patients with primary tumors that were drained by vessels leading to the lung would eventually develop lung metastases. Today, we know that both theories contain valuable truths, yet the full story of metastasis is much more complicated than either of the two proposed theories. Factors like the cancer cell's properties and the effectiveness of the immune system in eliminating the cancer cells also play a role in determining the success of metastasis. Unfortunately, many questions about metastasis remain unanswered till today. Understanding the exact mechanism holds an important key to finding a cure for advanced stage cancers. By studying both the genetic and environmental factors which contribute to successful metastasis, we can pinpoint ways to shut down the process. The war against cancer is a constant struggle, and scientists are hard at work developing new methods against metastasis. Of recent interest is immunotherapy, a modality which involves harnessing the power of the immune system to destroy the migrating cells. This can be done in different ways, such as training immune cells to recognize cancerous cells via vaccines. The growth and activity of the immune cells can also be stimulated by injecting man-made interleukins, chemicals which are usually secreted by the immune cells of the body. These two treatments are only the tip of the iceberg. With the collaborated research efforts of governments, companies, and scientists, perhaps the process of metastasis will be stopped for good. Begins
Okay, so that was a great video, a little bit too cartoonish, but I think it has a great way to show metastasis and in a simple way.
Begins
Okay, so now to finish the class, I just want to show you the different type of ways that the cells can move. These are fibroblasts that are moving, yeah. So not only cancer cells are moving, all those cells that you can have single migration here. Yeah, so here they are migrating a 2D surface. Yeah, so in a culture you can make them grow. Move. And just to remind you, fibroblast is a type of cell of the connective tissue. If you study uh, anatomy and physiology, you know that there are different type of tissues, and this one is the one that is located in, that is the extracellular membrane, and is form collagen and other fibers that later we will talk more about it. So, but here the key point is, remember, fibroblasts are important, and those one display single cell migration. Sometimes, if you remember the video that we just watched, some cells can move displaying single cell migration. Um, There are different type of migration in addition to the one that we saw it. But before that, I'm going to tell you a little bit of the mechanism. So one of the mechanisms that it has been found to allow this single cell migration is the formation of these rich actin extensions. Yeah, actin rich extensions that you can form here, and that allow them to attach and deattach to grasp one region. The leading region is going to grasp and is going to, while the other region is going to detach and is going to move. Yeah, and a similar type of movement have been found in cancer cells. Yeah, so again, these are the steps. You need to establish a polarity where you have a leading edge and a rear end. You need polymerization of actin, that is the protein that is involved in the different movements, yeah, in the changes in shape of the cell. Then you need to find adhesion points, yeah. So here you find an adhesion point that is going to help the cell to create friction, and then you disassemble this the back, the addition in the back, and this is constantly happen during development and also happen during cancer cells. Okay, so the video, I found that it was very clear, but in the same way that it was showing the video, I just wanted to show you that this is not the exception. Here you can see a culture. In the same way that we have cancer lines, yeah, like the HeLa cells, we have many more cancer lines in these lines, yeah. Since the HeLa cells were formed, scientists start looking for different type of cancer lines that can be test different type of cancers. So, for example, this is a fibrosarcoma. If you remember, is a cancer originated from fibrous and connective tissue, and this is the name of the cancer line. And if you see here, they put the scientists put a tissue culture with small gaps here. Yeah, that they are very difficult to go through. And what they did is took those cancer cells and they label it with GFP. And if you see, they are able to go through all these very small spaces and remodel their cell shape and to go through those spaces. So in this case, this has been very useful to understand how the cancer move and how they get this ability to infiltrate capillaries, blood vessels, and to go inside or to go outside. Yeah, it's same thing that we watch in the video. Now we watch it with real samples. So here is an schematic how they make the cells move, because here you have a cover glass that have a chemosynthetic chemotactic gradient. So the cells feel the need to move to a specific region, and this allow them to move. So this is a model to study metastasis, to study all the migration of the cells, so that they can move from one region into another one. And remember that they can move here from the capillary wall, go inside, go outside, and move into different tissues. And remember that during cancer metastasis, the tumor penetrate through tight spaces, and these spaces are called interstitial spaces, and they require a deformation of the cell and its nucleus. Yeah, so as you can see here, so it's an amazing property that they gain after. So if you see different tumors get different mutations, and this allows to get different abilities. So this is why you can start with epithelial cells, but you can start transforming other pieces of or type of cells. And it has been shown that, and we will talk about that, tumor it can be formed by a different community of cells, and we will talk about that in following courses, in following classes. Okay, so here you see three different type of migration. You have here single migration. Single migration, and I showed you in the last time. You have migration of a group of cells. You have here a collective, a group of cells that are moving together, and as you can see, there are different organisms at different stages. I'm going to show you more a little bit about lateral migration at the lateral line in zebrafish, but here, for example, is the early stages, the process of the puberty of zebrafish, that if you're interested later, you can take developmental biology, and we just see how those processes are taking place very superficially because there are so many things happening there. And here you can see the ovaries of a fruit fly, and I have seen this type of collective migrations. They're classical of mammary glands, in type of breast cancer. Yeah, and it's unbelievable that you can observe those things in fruit flies, but this is a behavior that occur, and that when you study it from a cell biology perspective, you see that there are a lot of commonalities. So, for example, you have cell protrusions. Yeah, so actin, the cytoskeleton need to be reshaped. You have other processes like the activation of molecules and proteins like RAC, that we are going to talk about that. So the cellular machinery is always is acting. It needs to be remodeled, and there are physical cues like collagen fibers, myofibers, and the basal lamina. Yeah, so all these microenvironment allow and send signals that allow the cells to move. Now I'm going to talk a little bit more about the lateral line of zebrafish because I generally talk about fruit flies. So I'm going to tell you a little bit about zebrafish. So here is a movie that shows us the lateral line and how is forming. Here is the adult, yeah. Here you can see the eyes, and here you can see the lateral line, and the lateral line is a great model to study cell migration. Yeah. Why? Because you can, as zebrafish is transparent, and you can have many genetic tools. Here you can, for example, see the label only the lateral line and say how the cells move in vivo. Yeah, so well, with the HeLa cells and with the other tissues we were talking about in vitro system, here you have an in vivo system that you can have a lot of genetic tools, so you can see the interaction of tissues, the different signals. The lateral line is a sensory system, so you here have individual sensory organs, yeah, that are interacting in the surface of the body. And it has been found that different type of cancer display a misregulation in this process that mimic the cell migration that you can see in the lateral line of zebrafish. Okay, so again, there are many type of cell movements, and some of them you can find in during cancer, during the process of invasion. Hear about this, yeah, is the migration, that it could be single but also collective, yeah, that organisms are able to do that, yeah. So in cancer you can find, I have seen in one leg cells when you produce uh, backgrounds that you promote an oncogenic growth. uh, you can see sometimes cells moving inside, displaying single migration. Well, you can see on the top of the cell collective migration, and you say like, oh my god, but how this could happen? I'm going to show you an organism that can do that. Okay, so here, this each one of these dots that you see here are single cells, and they start interacting, and they start displaying a collective migration. So instead of behaving uh, as a single organism, they start behaving as a multicellular organism when they start forming here. Okay, and here they start moving as collectively, although in the beginning they were moving as a single organism. So you have heard about this many times. Let me show you in the beginning here when they were just uh, unicellular organisms, that there is a chemical signal that allows them to do this change because of the environment, to be more efficient, and now they can become have a collective movement. So this is a great model to study this transition between individual and collective. And I'm sure that you studied this in diversity of life, that there are different organisms that we are amazed of their abilities, yeah. And Dictyostelium is one of the most amazing, that it can help us to understand the transition between unicellularity to multicellularity, yeah, and how some organisms, they can, without they can still um, assemble and disassemble, and a tumor can definitely behave sometimes as a single entity, and sometimes it can also work as a collective group of cells that are moving. Okay, and here are the different stages. Now, these are again a cancer lines. So they take a tumor from somebody, and they find a way to study, to grow it in the lab. So let me show you here. Again, this is an in vitro experiment, and these are lines that can be very useful to study, for example, drugs, for example, how the cell moves. Yeah, and they have been studied for a long time. So rather than tell you that in vitro experiments or in vivo experiments or in silico experiments are better, is a combination of all of those. What give us a better perspective of what is happening. And you will see when we study treatments of cancer, same thing happen. Rather than saying chemotherapy is the best or surgery is the best or immunotherapy is the best right now, what we're finding is that combining combination of therapies is the one that is more efficient, and try to make it as selective for the patient. Yeah, personalize the treatment. It seems to show the best results. Okay, so I to finish this section, I told you that there are three general stages: the primary tumor, the circulation, and here you need different type of genes and different type of changes in the anatomy of the cells. And I show you that to move a tumor to move the cancer cells, it require invasive behavior, yeah, cell movement, and there are a lot of tumors can migrate individually, they can migrate collectively, and I show you that during development, all these different organisms that we study, all these model organisms show examples of cell migrations, and all of them can be found, or many of them can be found in different type of tumors. I show you also that this process of colonization is also very important. It also requires different genes. And in the following classes, we are going to cover more topics about that. But I just wanted to give you a very general perspective of what is happening. So remember, here you will need initiation genes, and you will need that you have these four changes, yeah, and one of them was the mobility of the cells. In our class, I will show you more about angiogenesis. You have progression genes that allow to, if you remember those green cells and the video that I showed you, how the cell have to squish between capillaries and how they need to be able to survive. And finally, the virulence genes, and that they confer the potential for colonization. Yeah, that we're going to talk about that. And in the future, we will talk about this word that the second part of the course will talk about the importance of the environment, and in particular of the microenvironment. And we're going to talk about cancer stem cells, yeah, and also later we can talk about this topic of exosomes. Sorry. So and there is a lot of topics that we will cover in a lot of detail, and other ones that I will tell you a little bit during the class. And other topics, for example, these exosomes is a topic that is in development, but we know that is becoming very important. Vesicles are becoming very important. But anyway, I hope that you enjoyed this part of the class. Later, I will tell you now about the genetic basis of cancer, and I hope that you had a lot of fun. Bye.